Hello, fellow neuroscience enthusiasts. This video is another installment of the Brain Preservation Foundation's Neuroscience Journal Club, in which we briefly review the results of a selected paper and discuss its relevance to the ongoing debate regarding brain preservation. Today's paper is entitled Interregional Synaptic Maps Among Engram Cells, Underlie Memory Formation by Choi et al. It was published in the journal Science on April 27, 2018. A link to the paper is listed below. This is a truly fantastic paper that employs some of the most advanced genetic techniques to test a central prediction of today's main theory of how the brain encodes memories. Unfortunately, I will only be able to cover a fraction of what is in the paper, so please do try to read the full paper yourself. Also, please note that although I will try my best to accurately relate the research, it is always possible that I may misinterpret something crucial. If I do, please let me know in the comments and I will try to correct it. This paper is just one of a plethora of papers we will be covering that seek to determine how memories are physically stored in the brain. Many neuroscientists, stretching all the way back to Donald O. Hebb, have suggested that collections of interconnected and thus coactivating neurons are likely to be the main representational units in the brain. These collections of neurons are variously referred to as cell assemblies, neuronal assemblies, neuronal ensembles, and Engram cells, which is a term used when an ensemble is associated with a particular memory. As this figure from a 2005 review by Kenneth Hayworth suggests, a noisy or ambiguous sensory input succeeds in activating a particular cell assembly out of a range of competing cell assemblies. Its activation provides the clear and unambiguous representation necessary to drive subsequent cognition. Crucially, this type of pattern recognition is dependent on strong excitatory synapses among the cells of the cell assembly, which perform a type of neural network pattern completion. This cell assembly drives the activation of other cell assemblies, which in turn drive the activation of yet other cell assemblies. As Harris suggests in his paper, the evolution of this phase sequence is the hypothesized substrate of internal cognitive processes. Many other authors have stressed how this cell assembly or neuronal ensemble viewpoint provides a key link between the study of biological brains, where we can now record neuronal ensembles using techniques like two-photon calcium imaging, and artificial neural network theory, where such neuronal ensembles are predicted to be a product of the attractor dynamics in the networks. Let me briefly summarize this neuronal ensemble theory of cognition and memory in perhaps its strongest terms. Every active brain representation, either of an aspect of the external world or of an internal mental state, is associated with a particular ensemble of neurons firing in a particular brain region. Such neuronal ensembles are simply a reflection of the region's excitatory synaptic connectivity. Neurons within a particular ensemble share a larger number of strong excitatory synapses with other members of that same ensemble than they do with neighboring neurons that are not part of the ensemble. And I note here that this is the main prediction of the Choi et al. Paper that we will be going through today. The set of ensembles, or also known as attractor states, available in a particular brain region may initially form as random groupings of interconnected neurons that happen to strengthen their interconnectivity due to transient increases in their neuronal excitability. These ensembles take on meaning as learning mechanisms associate them with particular sensory or internal states, again via synaptic strengthening. Long-term perceptual and sensory motor learning involves the strengthening of synaptic connections between ensembles in low-level sensory regions and ensembles in high-level association regions, for example, V1 to IT cortex. The formation of episodic memories involves the formation of associations between multiple widespread cortical ensembles. This is accomplished by strengthening the synaptic connections of each of these cortical ensembles with a common ensemble, for example, in the hippocampus. And more complicated representations, such as sequences, are hypothesized to be formed by strengthening synaptic connections between sequentially active ensembles, which are then chained through by oscillatory brain mechanisms. There is so much more I would like to say about how an understanding of neuronal ensemble dynamics is key to understanding brain function, but I will wait till other videos for that. For now, let me suggest that interested viewers read this 2016 review by Holtmat and Caroni, which provides an excellent and up-to-date review of all of these concepts. I highly recommend reading it. Now back to our paper for this journal club. Choi et al.'s experiment explicitly tested the prediction that neurons within a particular ensemble, or memory engram in this case, share a larger number of strong excitatory synapses with other members of that same ensemble than they do with neighboring neurons that are not part of that ensemble. Since Choi et al.'s experiment tagged a neuronal ensemble that was associated with a contextual fear memory, they used the term Engram, and I will switch to that terminology for the rest of this video. So Engram equals a neuronal ensemble associated with a particular memory. Their experiment involved activating and then labeling a particular engram in the hippocampus, and then counting the number of excitatory synapses those neurons shared with each other relative to what they shared with so-called non-engram neurons, that is, nearby neurons that were not part of the actively labeled engram. With that background, let's try to read through the Choi et al. 2018 abstract. Here we go. Memory resides in engram cells distributed across the brain. However, the site-specific substrate within these engram cells remains theoretical, even though it is generally accepted that synaptic plasticity encodes memories. Now, this is what their experiment strongly supports. We developed the dual-eGRASP, GRASP standing for Green Fluorescent Protein Reconstitution Across Synaptic Partners. Okay. We developed the dual-eGRASP technique to examine synapses between engram cells to identify the specific neuronal site for memory storage. We found an increased number and size of spines on CA1 engram cells receiving input from CA3 engram cells. Now, this is the key result of their paper, showing that there is a larger number of strong excitatory synapses connecting engram cells. In contextual fear conditioning, this enhanced connectivity between engram cells encoded memory strength. That is, the enhanced connectivity was correlated with the behavioral strength of the contextual fear memory. These results indicate that enhanced structural and functional connectivity between engram cells across two directly connected brain regions forms the synaptic correlate for memory formation. Exciting stuff. Let's see how they did it. The Choi et al. Study involved the contextual fear conditioning of mice. A mouse is placed into a particular spatial context, a unique cage, for example, and its feet are shocked, giving rise to a memory that will make the mouse fearful of that particular cage. It is well known that the hippocampus is involved in encoding such a contextual fear memory. Structures in the hippocampus somehow record the association between the aversive shock and the representation of the cage the mouse was in while being shocked. Choi et al. Used the doxycycline c-fos system, which you may remember we covered at length in a previous journal club. to label neurons that were active while the fear memory was being encoded. Here is the fear conditioning timeline of the Choi et al. Experiment. Dox was injected into the mouse during a brief anesthesia by isoflurane, and 2 hours later, the mouse underwent contextual fear conditioning. 2 days later, the mouse was euthanized and perfused for imaging. 14 days prior to fear conditioning, a set of viral vectors, AAVs, were injected into the CA3 and CA1 regions of the mouse's hippocampus. These 4 viral vectors were injected into the CA3 region of the mouse's hippocampus. These 2 viral vectors ensured that neurons in CA3 that were active during the fear conditioning would express the yellow pre-eGRASP protein. These 2 viral vectors ensured that a small subset of other randomly selected neurons in CA3 would express the cyan pre-eGRASP protein. At the same time, 4 more viral vectors were injected into the opposite hemisphere's CA1 region of the hippocampus. These 2 viral vectors ensured that neurons in CA1 that were active during the fear conditioning would express the red fluorescent protein M scarlet and would express the protein post-eGRASP. And these 2 viral vectors ensured that a small subset of other random neurons in CA1 would express the infrared fluorescent protein IRFP670 and the protein post-eGRASP. This is a diagram of a cross-section of the hippocampus. Axons from the neurons in the CA3 region make recurrent connections with other CA3 neurons and send axons to make synaptic connections onto CA1 neurons. Other studies have shown that hippocampal memory formation involves, among other things, the strengthening of synapses from these CA3 neurons onto these CA1 neurons. The CA3 neurons of the left hemisphere also project to the right hemisphere hippocampus via the fornix bundle. And they make synapses onto the CA1 neurons of that opposite hemisphere. The Choi et al. 2018 paper is studying these synaptic connections made from the CA3 neurons of one hemisphere to the CA1 neurons of the opposite hemisphere. I'm going to switch to a schematic depiction of this to make the logic of the experiment clearer. Here I am showing the CA3 cells of one hemisphere with their axons projecting to the other hemisphere, where they make synapses onto that hemisphere's CA1 cells. In the native animal, none of these cells or synapses are labeled, so it is impossible to image which neurons are synapsing on which. When the viruses are injected, a small subset of random neurons in CA3 will be infected and start expressing cyan pre-eGRASP. And a small subset of CA1 cells will be infected and start expressing IRFP670 and post-eGRASP. Now, the IRFP670 will make these CA1 neurons glow in infrared light in the fluorescent imaging. This is shown as a purple coloring here. The crucial thing to note is that the cyan pre-eGRASP will combine with the post-eGRASP only at the synapses between these 2 types of cells, thereby causing these synapses to glow cyan in color. This is what the GRASP technique is all about, labeling only those synapses where a pre-eGRASP expressing neuron synapses onto a post-eGRASP expressing neuron. As I will show later, these synapses will show up as small cyan glowing dots in the light micrographs. Now, during the contextual fear conditioning, those CA3 cells that are active will start expressing yellow pre-eGRASP. And those cells that are active in CA1 will start expressing M scarlet and post-eGRASP. The M scarlet will cause the active CA1 cells to show up as red in the fluorescent images. These yellow pre-eGRASP labeled CA3 cells and red labeled CA1 cells are the Engram cells that were active during the fear memory formation. Let me pulse these cells again. These are the Engram cells that fire together during memory formation and during memory recall. Crucially, the synapses between the CA3 Engram cells and the CA1 Engram cells will be labeled in yellow in the fluorescent images because the yellow pre-eGRASP will combine with the post-eGRASP at those synapses. I will pulse these synapses now. Also, any synapse made from a CA3 Engram cell onto a CA1 non-Engram cell expressing post-eGRASP will also be labeled in yellow in the fluorescent images. I will pulse this one now. And any synapse made from a CA3 cell expressing cyan pre-eGRASP onto a CA1 Engram cell will be labeled in cyan in the fluorescent images. I'll pulse this one now. Now, in the actual experiment, only CA1 is imaged, and only those cells and synapses that are labeled with fluorescent proteins will be visible. So we'll actually see something more like this. But from this, we can clearly distinguish the four classes of synapses. Here is a figure from the Choi et al. Paper that shows these four classes of synapses. A cyan synapse on a purple CA1 neuron is a connection from a non-engram cell to a non-engram cell. A yellow synapse on a purple CA1 neuron is a connection from an engram cell onto a non-engram cell. A cyan synapse on a red CA1 neuron is a connection from a non-engram cell onto an engram cell. And crucially, a yellow synapse onto a red CA1 neuron is a connection from an engram cell onto another engram cell. If memory engrams are collections of cells that fire together because they share more excitatory synapses, then these engram to engram synapses should be larger and more likely than other synapse types. This is a projection of an actual 3D fluorescent volume image of CA1 neurons from the Choi et al. Paper. The red neurons are engram cells in CA1, and the gray neurons are non-engram cells in CA1. Note that the gray color in these fluorescent images corresponds to the purple neurons in our previous schematics. Choi et al. Used the 3D fluorescent volume, which was in 3D, to reconstruct the dendritic shafts and label the spiny synapses as shown here. And zooming in, one can see how each yellow or cyan labeled synapse was associated with a particular red or gray neuron. And here is the key result. When the relative spine densities of each class of synapse was compared, they found that there were significantly more engram to engram synapses than would be expected based on the other categories. They also found that the sizes of engram to engram synapses were significantly larger than the other classes. This harkens back to our last journal club covering the Kasai et al. 2003 paper, where we reviewed evidence that the size of a spiny synapse is correlated with its functional strength. And the Choi et al. Paper even showed that this spine density and size difference was greater in mice that underwent a stronger fear conditioning. Reading from their conclusions, our finding that synaptic populations that fired together during memory formation showed the strongest connections demonstrates that classical Hebbian plasticity indeed occurs during the learning and memory process at CA3 to CA1 synapses. It is possible that cells with higher connectivity are allocated together into a memory circuit, in contrast to enhanced connectivity after learning. However, the allocated cell number remains constant regardless of the memory strength, whereas the connectivity is significantly enhanced with a stronger memory. This finding indicates a significant contribution of post-learning enhancement over the predetermined connectivity. The relationship between memory strength and synaptic connectivity suggests that these specific connections between engram cells across two directly connected brain regions form the synaptic substrate for memory. Now, this particular study addressed a very specific question regarding the encoding of memory, specifically whether memories are stored as the strengthening of synapses among ensembles of neurons. The paper and this short video certainly do not address the specifics of how the hippocampus interacts with the rest of the brain to store and retrieve specific memories. For viewers interested in that, I would recommend these two papers. Rolls and Kestner's classic paper, A Computational Theory of Hippocampal Function, and John Listman's 2015 review, which has an extensive section on the hippocampus, including a model of how it can store and replay sequences. It takes neuromodeling papers like these to really answer the question of what it means to say memories are stored in synaptic connections. But it takes experimental papers, like the one we just reviewed by Choi et al., to test the predictions of these theoretical models to see if they are indeed on the right track. Now let me very briefly address the relevance of this particular paper to the ongoing brain preservation debate. First, this Choi et al. 2018 paper is yet another primary research paper that strongly supports the idea that memories are stored by the strengthening of synaptic connections between particular sets of neurons. And the paper shows that there are clear structural changes to these strengthened synapses that can be read off by modern imaging techniques. The Choi et al. Paper and the many others that we have referenced in these videos, argue that we do have some idea how memories are stored in the brain. And these papers argue that there is reason to believe that connectome preservation would be sufficient for the preservation of memories, although the evidence to date is certainly not proof of this. This result, and the many others like it, should give pause to anyone that casually dismisses aldehyde-stabilized cryopreservation as a means to preserve the information content of a brain for long-term storage. Recall that ASC, since it is based on glutaraldehyde perfusion fixation, is known to preserve the synaptic connectivity of a brain, and is known to preserve ultrastructural features of synapses that are correlated with their functional strength. Choi et al. Used genetically targeted fluorescent labeling of synapses, so they were able to easily determine the volume of each spiny synapse using light microscopy, showing that their numbers and sizes were correlated with the behavioral strength of a contextual fear memory. But it should be obvious that these same structural changes to synapses would also have been visible if the tissue volume had been preserved by ASC and subsequently imaged using 3D electron microscopy. And 3D electron microscopy has the advantage that every neuron, axon, dendrite, and synapse can be mapped simultaneously in a single experiment. This raises the possibility that 3D electron microscopy might be able to map out all of the neuronal ensembles in a brain region, essentially mapping out the vocabulary that that region can support. Mapping out the synaptic connectivity, for example, in a primary sensory area, would effectively ground each neuronal ensemble to a sensory feature. And mapping out the synaptic connectivity among ensembles could allow us to determine how cognitive operations are sequenced so that they can give rise to intelligent behavior. I intend to explore these ideas in more detail in future videos. Thank you for listening, and thank you for keeping an open mind regarding what advances in neuroscience might bring in the future. These videos are designed to spark conversation and debate, so please feel free to leave comments below. I look forward to reading them.